Howdy, today on Flippin Science, looking at enzyme function and the induced fit model. Thanks to Sam Moyle for producing this PowerPoint. So science understandings we're going to look at. Enzymes are specific for their substrate and increase reaction rates by lowering activation energy, and describe the induced fit model of enzyme substrate binding. So hopefully you know a little bit about chemical reactions. So reactants produce products. Reactants might combine, they might break up, or they might recombine, and you get products being produced. It could be a single reactant making multiple products. It could be multiple reactants making multiple products. It could be multiple reactants making single products. In chemistry, we can increase the reaction rate by using a thing called a catalyst. So what that means is we can make more C and D faster from A and B, and when we do that, we're increasing the reaction rate. Different catalysts work differently. So in your car, you'll have a catalytic converter. That takes some of the dangerous gases produced from your car engine and turns them into safer gases. There's expensive metals in there, things like platinum and palladium, and that's why they're being stolen at the moment. But catalysts, they help. They reduce the activation energy required for a chemical reaction to occur. So if there's less energy required, then you get faster production of turning reactants into products. Biology, same idea, except we've got some different terminology and some different things happening slightly. Instead of saying reactants, we say substrates. So substrates form products. And our catalysts in biology are often biological, and a biological catalyst is an enzyme. And an enzyme is a protein that's folded in a specific shape to increase the reaction rate. In chemistry, we talk about reaction rate in terms of you need your molecules that are reacting to collide in a specific orientation with a specific amount of energy. Enzymes provide the orientation. They provide the right direction for the reaction to occur, and that's how they speed up the reaction rate. So you don't need as much energy to produce as much product. So you get more product produced in less time, and that increases the reaction rate. So just restated, enzymes are biological catalysts. So you've got thousands of reactions occurring in your cells all the time, and they happen very slowly if you don't have catalysts. So we have enzymes that are catalyzing these reactions. They're speeding up the chemical reactions by reducing the amount of energy required for the reactions to occur. Enzymes are proteins, and they're produced by your cells, and a lot of the instructions in terms of genes on your DNA are just talking about making specific enzymes in specific shapes. Each enzyme is specific to a particular reaction. So you have certain substrates producing particular products. Each enzyme is specific, and the reason for that is the shape of the substrates and products is going to be different before each reaction that's occurring. Mostly enzymes are catalyzing reactions inside the cell, so they're intracellular, but some happen outside the cell, maybe in the extracellular fluid, so they're extracellular enzymes. Key idea, and this is the same in biology and chemistry, catalysts aren't consumed in the reaction. So any catalytic converter in your car, once it's set up, it's going to work for a long time. Same idea with enzymes. Once you've made an enzyme, it can catalyze many reactions really quickly. So you're making a specific amount of enzyme to catalyze reactions. But you don't need much because each enzyme can be reused many times. So you make some enzymes and they speed up reactions, but you don't need to have huge numbers of those enzymes. Restating something that we stated previously, the 3D structure of a protein is very important for its function. The tertiary or quaternary shape of a protein stipulates the function of that protein. And that's why enzymes only do what they're made to do, what they've evolved to do, which a reaction that they're catalyzing. Enzymes are globular proteins with a region that has a specific shape for binding purposes, and we call that an active site. So here we have collagen, which is fibrous. So it's lots of fibers of collagen that wind around each other. That's different from a globular protein. So globular proteins, they're globular. They're kind of ball-shaped with an active site. So here's my globular protein, here is my active site, and here's my substrate. So the substrate comes into the active site changes shape a little bit, and then you get your products being produced from that. This is a schematic model. Here is a molecular model. This is catalase. Catalase is an enzyme which breaks down hydrogen peroxide. Hydrogen peroxide is a very reactive molecule, and you don't want too much of it in your body. So you make catalase, which breaks it down. The active site is down here. So the hydrogen peroxide comes into the active site down here in catalase. It gets the active site changes shape. It pulls apart the hydrogen peroxide into water and oxygen, and you get water and oxygen coming out. from the active site. And here's my little animated arrows showing you where those active sites are. So the substrate here binds to the active sites here. When the substrate binds to the active site changes shape slightly, and that leads to the production of your products. So we've got our word complementary popping up again here because our substrate is complementary to the active site on the enzyme. So only the right shape of substrate will fit into the active site of the enzyme, and that will lead to the reaction occurring. That means there's specificity. So only a specific enzyme can catalyze a specific reaction. Here's our animation showing that. So our active site here is complementary to our substrate here. Let's just talk about the induced fit model to finish off with. So when the enzyme and substrate join together, the active site changes shape slightly. And what that does is it puts a stress on the bonds in our substrate. In chemistry, we'd say our reactant. It puts stress on the bonds, and what that can do, it can break the bonds and form new products, and then that's what's released from the enzyme. Sometimes the active site changes to force different aspects of the reactants of the substrates together to make a product. Either way, we're stressing bonds and we're making new bonds. We might be breaking bonds, and that leads to the production of our product. So it's that change in shape we call the induced fit, because we're inducing a fit. The shape is changing slightly to make the fit a little bit better, which leads to the stress on the bonds, breaking and reforming bonds to produce the products. So before I said in chemistry, we talk about reaction rate being moderated by things like how much energy the reactants have and whether the reactants are colliding in the correct orientation. By having this active site here, we're providing the correct orientation for the reaction to occur, so we don't need as much energy for the reaction to occur. We don't need as much smacking into each other of the molecules to make our products because we're providing a space for that to happen. So that means less energy is required, and that speeds up the reactions. So here's some animations. So here we have our substrate, here we have our enzyme. We can see our substrate goes in, that changes the shape of the active site a little bit, and we get our products being produced. Over here, same idea. So here's our substrate. It's going into the active site. The active site is changing shape a little bit. That puts stress on some bonds, and that produces our products over here. So our check. So enzymes are specific for their substrate. They increase reaction rates by lowering activation energy. And you need to describe the induced fit model of enzyme substrate binding. So today at Flippin Science, we looked at enzymes and the induced fit model. That's it for Flippin Science today. See ya.